So DNA replication. We've talked about it before, but we just said you have a chromosome and it duplicates. It's an exact copy, right? These are the sister chromatids, if you remember that. We talked about this before any sort of cell division. Any kind of cell division, we're gonna make more cells, we need more DNA. We even talked about it in the cell cycle, right? It went through this process before mitosis. There's the S phase, the synthesis phase, where we made more DNA.
So if you remember, it looked just like this. During the S phase of the eukaryotic cell cycle, that's when we took that one chromosome and duplicated it. Now we're just going to zoom in on the duplication part and figure out how sister chromatids are made.
Now we're going to take a look at these chromosomes really closely. We're not going to get out of the scale where you've seen it before, right? You've looked in a microscope and seen chromosomes, or these groups of them in those onion root tip cells. But now we're going to zoom in to the molecular level. And things get a little fuzzy at that point. When we do, we use a variety of models. So there's all kinds of them. But you'll notice that a common theme is the shape. It's often referred to as a double helix. So that double helix shape just means it's a spiral, that's the helix. And there are two molecules of DNA here. So they have this backbone here in black, right? There's one, and then there's this other one over here. And they're held together in the middle by hydrogen bonds between those nitrogen bases. So you should have some background on that from learning about the structure of DNA itself. And that structure is important to understand how DNA copies itself. As we get into a little bit later, then the shape becomes important. It's a flexible molecule. There's that double helix again. And this one doesn't split apart, but we can unwind it. And that's going to be one of those things that DNA is going to have to do when it goes to being replicated, is actually unwind. We'll use some paper models as well to represent DNA. So we got all these long strings of letters, and those go together. Oh, we got that one turned over. And they form these complementary bases inside. Complementary base pairing, really important concept that you understand that. And that's that idea that the G's go with the C's on this side, and the A's go with the T's. Pretty simple rules. G with C, A with T. But don't think of it as matching, right? It's not that they match, it's that they're complementary. Like your shirt might complement your pants. They're not the same, but they complement one another. When we copy DNA, we don't do it the same way that we copy things in person. So if I had a drawing and I wanted to make a copy of it, well, I just draw another one, right? Or I take this one and I put it in the copy machine and I get this one out. So we got copies. That's not how DNA works. But remember, DNA is not a single helix, it's a double helix. There are two pieces here, two molecules in a big complex. So when we go to copy DNA, we actually first need to rip it in half. And what we do is we say, I have this half, you give me the other half. And it knows just how to fill that in. And it does the same thing for the other side. You have half, it gives you the other half. It's a weird system of copying. Certainly not the way we think of copying, but it's all about complementary base pairing. If you have half, I'll give you the other half.
Well, the basics of DNA replication are not too difficult. Here we have our double-stranded DNA. The first thing that needs to happen is to separate those two strands. Remember that everything here is held together by covalent bonds along this sugar phosphate backbone. Same over here, but it was hydrogen bonding that held these nucleotide bases together, and that was fairly easily separated. An enzyme will come in and match complementary bases. Remember those rules, pretty simple. Thymine and adenine are always complementary, and cytosine and guanine. So the one strand codes for the other. One term given to this is semi-conservative replication. Fairly simple. Again, two steps. Unwind and unzip first. We talked about unzipping before, but remember it is wound up in this double helix, so we need to do that too. That exposes each of the strands, and then we just pair up complementary bases using the enzyme DNA polymerase. What we end up with then is two identical molecules, maybe more accurately, two identical double helices of DNA, one with the old strand, the blue, and a new strand, the yellow. So each of them have an old and a new. We can see the details of that over here on the right, where complementary bases are being added in yellow and the old ones in blue. I also notice that these are moving in opposite directions. In one strand, synthesis is taking place that direction, and in the other one, it's going in an upward direction, which we'll come back to later.
Well, that's a good basic overview of DNA replication. Let's go back and add some details now. Let's learn some terminology. Let's talk about the enzymes involved in the process that shepherd this along, and let's learn some of the rules and complexities of how it all works.
Now, DNA replication is a fairly universal process, but it does happen a little differently in prokaryotes and eukaryotes. So let's start here with binary fission and look at the nucleoid region. Remember, there's a single ring of DNA, and that gets replicated, and then the cells split. Well, how does that happen? Well, we have double-stranded DNA in one big ring. We would have to split those apart first. Then we could get an enzyme, DNA polymerase, to come in and start on one of those strands, start synthesizing the new strand. Complementary base pairing, adenine with thymine, guanine with cytosine, all along the way for hundreds of base pairs forming those covalent bonds. But when we reach the end, we can't stitch the beginning to the end. So we need something new. Now, the other loop is going to be synthesized in just the same way. What we need to link up the beginning to the end is DNA ligase. Because see that little gap right there? Right, that little bit of yellow there, right there. So that needs to be hammered together, right? That's what ligase does. Hammers it, it's nailed together, and we're done. I'm not sure if it's occurred to you or not, but where does the energy come from for this, right? We're making brand new molecules. That's synthesis. That should be an endergonic reaction of some sort. So do you remember our good friend ATP over here? Well, it has cousins, GTP, CTP, and TTP. So this was adenosine triphosphate. This is guanosine triphosphate, and so on. So all those nucleotide bases, and ATP is a nucleotide base, carry 3 phosphates. Remember, those bonds have energy in them, and when they're linked on to the next one by DNA polymerase, those bonds break, release energy, and allow the synthesis to take place. There it is.
Here we have our double-stranded DNA. I straightened it out for us so we don't have it all twisted up, make it a little bit simpler to look at. We know that the first step is that DNA needs to be split. That's done by helicase and a partner enzyme called topoisomerase. So that splits it apart so we can access the nucleotides in here and lay down complementary bases. The next enzyme that acts is DNA polymerase. It's the one that's going to pull in the free nucleotides scattered around inside the nucleus and line them up with their complementary bases on either strand. So one specific rule about how DNA polymerase works is that it only moves one direction on the DNA. Specifically, it moves from the 3' end to the 5' end. So let's look at that one. Over here, we have the 3' end. We can tell because this sugar, the way it's arranged, this is the 3' carbon on the sugar. Since DNA is antiparallel, then this strand points the opposite direction, and that's the 5' end. So the other end of the DNA is the opposite. We have the 3' end and the 5' end.
Well, the term antiparallel may sound really foreign. It's actually a familiar concept if you've driven down the road. See, these two run parallel to each other, right? But in opposite directions. You just got to follow the rules. Stay on the right side of the road, no one gets hurt.
So DNA polymerase is really the star of the show, but it's a very picky enzyme. It only works in specific ways. We talked about it moving from 3' to 5'. That's one tricky thing. But the other one is it can't just start here in empty space. it actually needs to recognize a little piece of double-stranded DNA. And so there's another enzyme called RNA primase that lays down a small piece of RNA, call it an RNA primer, and it is complementary to the DNA. That is actually what allows DNA polymerase to land here, and it's actually going to start right after this. All right, back to that more in a bit. One more wrinkle that's a little complicated. In eukaryotic organisms, instead of just a simple DNA polymerase like we have in bacteria, it actually moves in this mega complex. So helicase comes and splits the DNA, and there's actually two sides of the complex, each with their own DNA polymerase. All right, you may see the problem with this already. This DNA polymerase is moving the correct direction, 3' to 5', but in this one, it's going backwards, and this enzyme is not built special enough to actually operate in a backwards fashion. Fortunately, there's an easy solution. Well, sort of easy. DNA is flexible, and so we can actually take, this is the 3' end, right? And loop this around like this so that this polymerase is moving from 3' to 5' on that loop. Now, I'm not talented enough to hold that in position, so I'm just going to leave them this way and allow you to do the mental gymnastics of looping that around. But that's complicated. It also means that my RNA primase needs to lay down another RNA primer, and it's going to do that a little ways up the road so that this DNA polymerase can start here. When the whole thing moves further down, it will even lay down another one way down here. So this strand over here is easy, right? It just moves 3' to 5' the entire way. That is the leading strand. This one is called the lagging strand. All right, so the process is really not all that complicated once we get those details down. Polymerase comes in. I always think of it kind of like a cheerleader spelling out the team's name, but in this case, it's the complementary bases that it's calling for. So when it runs into this one, it's going to say, give me an A. A. And it comes along to the next one and says, give me another A. And we'll just complete this. Keeps doing the same thing as it moves along. And we're done. You'll notice that this new strand constructed is identical to this old strand. Again, semi-conservative replication, old strand, new strand. One of the things that DNA polymerase was doing along the way was to actually go in and make those covalent bonds. Still hydrogen bonding, holding these two strands together. Now, while this was happening, and this DNA polymerase was laying down this strand, remember this strand was looped over on itself, so this DNA polymerase was moving the opposite direction, and I'll picture it over here, laying down new bases on this one. So we're done with this segment, and this is called an Okazaki fragment. So it's a piece of DNA, not complete yet. So once this whole complex, as you remember, moves down this direction, as this is being synthesized, then this is being made in the opposite direction. So let's fill in that gap. And we're done with that one. So now we have one fragment here, another fragment here. At this point, then this primase, or this primer, is going to go away, leaving a bit of a gap here. And a different polymerase is going to come in, a separate one, and actually fill that one in. It's going to say, give me a G, give me a T, give me a G. All right, so it's filled in that little gap where the primer was, making these bonds. We still have one more problem, which is that this and this don't have a bond. There's no covalent bond holding them together. there's a bit of a gap here. And so there's one more enzyme at work. So that enzyme is DNA ligase. And it's going to come in, and its job is to make that covalent bond. And now all this is held together by covalent bonds. Now you might think that I have more to go here, but actually I don't. I'm done. Yeah, but what about these primers, you might ask? Well, this is a flaw in the system. This primer cannot be replaced. There's no polymerase that eukaryotes have that can come in and replace that. Same over here on this 3' end. And so every single time that our DNA replicates, there's a little bit on the ends that's left undone. And the solution to that is to add DNA on the ends, so-called junk DNA, into segments we call telomeres.
And here's a picture of those telomeres. On the ends of each chromosome there, that bit in red, is this junk DNA, the stuff at the end. And as cells get older and we undergo more and more divisions, those telomeres get shorter and shorter, which may have a lot to do with human aging. Well, there you have it. That's the nuts, bolts, and everything in between for DNA replication.